Good morning and good afternoon to those of you joining from China. It is an honor to be invited as the international speaker for this SIROmics webinar. Thank you to the BGI team for organizing it, and thank you for the opportunity to share a perspective from Bucharest. Before we go further, a word about the lens I am bringing to this talk. I am not a microbiologist. I'm a medical geneticist. My day-to-day work is constitutional disease, prenatal diagnosis, and oncogenetics in a regional genetics center that runs clinical NGS as a routine service. Through our research platform, my team also works in metagenomics and breathomics. So why is a geneticist opening a talk about infectious disease sequencing? Because infectious disease, when you look at it through a sequencer, is a genomic story. It's a contest between a host genome and the pathogen genome, and the winner of that contest determines the clinical outcome. The technology that reads both sides of that contest is the same technology, NGS, next generation sequencing. Clinical genetics has something that clinical infectious disease NGS is still building, a mature translational toolkit. We have learned what sample integrity means and when reads cost real money. We have learned that variant calling sometimes fails. We have learned how to write a clinical report that a non-geneticist can act on. The argument of this talk is that those same principles apply almost without modification when we point next generation sequencing at infectious disease. Two definitions worth aligning on, since the audience here is mixed across laboratory disciplines. Metagenomics, NGS or mNGS, is a hypothesis-free sequencing. You take a clinical sample, extract all the nucleic acid in it, and sequence everything. The pipeline tells you what is there. You don't need a list of suspects in advance. This is the fundamental departure from polymerase chain reaction PCR, from multiplex panels, and even from targeted NGS panels. Those tests answer the question, is the pathogen I asked about here? Metagenomics NGS answer a different questions. What is here? Targeted NGS sits between the two. You enrich for organisms on a curated panel before sequencing. Faster, cheaper, lower limit of detection on listed targets, but blind to anything outside the panel. The workflow is the same backbone you already know: sample, extraction, library preparation, sequencing, bioinformatics report. What makes infectious disease sequencing distinctive is what each of those steps has to handle: extreme host backgrounds, environmental contamination, no positive control to anchor against, and the clinical decision waiting at the end of it. The rest of this talk is about how that workflow either delivers or fails to deliver at the bedside. When does it actually make clinical sense to send metagenomic and NGS? 6 scenarios cover most of the routine practice. First, culture-negative infections in critical sites, central nervous system, deep tissue, endocarditis, where the patient is on empiric antibiotics and conventional workup has gone silent. Our anchor case today is exactly this scenario. Second, the immunocompromised patient with an atypical presentation where the differential is broad and includes opportunistic organisms that cultured poorly. Third, suspected zoonosis or imported pathogen, patients who have traveled, who have had animal exposure, where targeted testing requires you to guess in advance which organism to look for. And fourth, the fastidious organism, mycobacteria, fungi, intracellular bacteria, where culture takes weeks if it works at all. Fifth, failure of empiric therapy when the clinician is changing their mind about the diagnosis. Metagenomics NGS reframes the case rather than confirming the original hypothesis. Sixth, outbreak mode investigation of unknown pathogen, where epidemiology is moving faster than targeted assays can be developed. What unites these scenarios is decision pressure under uncertainty. Metagenomics NGS earns its place when conventional tests are silent and the clinical clock is running. Look at this chart. The descending line is the cost of sequencing one human genome on a logarithmic scale from 2001 to 2022. It has fallen by roughly 5 orders of magnitude. The flat line is the median time it takes in routine clinical practice from the moment an infectious disease patient is sampled to the moment a clinician acts on a sequencing-based results. That line has barely moved. This is the asymmetry that shapes the rest of my talk. The technology has won, the translation has not. A concession, the debate about whether next-generation sequencing belongs in clinical infectious disease practice is over. The technology is mature, we have peer-reviewed evidence, regulatory pathways, established clinical indication, validated workflows. Metagenomics NGS is no longer experimental. The honest conversation in 2026 is no longer about whether it works, it's about how we deliver it reliably, fast enough to matter into a clinician's decision at the bedside. That gap between a sequencer that has won and a bedside that has not yet received the benefit is what we have to close. And the gap is not made of technology, it's made of workflow, interpretation, and trust. It's made of 3 filters. The structure of the rest of the talk. Between the moment a sequencer finishes the run and the moment a clinician acts on the result, there are 3 filters. Each removes a specific kind of noise, and each in a real clinical program can fail. The first filter is physical, in the wet lab. It removes host DNA, contamination, and sample quality noise. The second is computational, in the dry lab. It removes the database level noise, false hits, miscalls, signals from organisms that are not really there. And the third is cognitive, at the bedside. It removes causality noise, the gap between detecting an organism and concluding that this organism is actually causing the disease in this patient. A program is only as strong as its weakest filter. We will walk through all 3. I want to introduce a patient. This is a composite case from a real clinical experience, and we will return to him 4 times, once now and once inside each filter. He is 67 years old, type 2 diabetes, otherwise reasonably well. He arrives at the emergency department with 5 days of progressively worsening headache, fever, and confusion. His family mentions a brief gastrointestinal illness about a week earlier. In the emergency department, empiric antimicrobial therapy is started immediately: ampicillin, ceftriaxone, vancomycin, and acyclovir. Note the ampicillin. In patients over 50 with diabetes, Listeria coverage is standard of care. All of this is started before the lumbar puncture is completed. The lumbar puncture shows a mild neutrophic pleocytosis, normal glucose, slightly elevated protein, negative Gram stain. By day 3, the conventional workup is silent, bacterial cultures negative, herpes simplex virus PCR negative, the multiplex meningitis encephalitis panel negative. Worth pausing here, that panel does include Listeria as a target, so the negative results itself a clinical clue, likely a reflection of bacterial DNA suppressed below the panel limit of detection by 36 hours of empiric antibiotic pressure. Hold that thought. The patient is improving slightly, but not clearly. The clinical team asks, should we send metagenomics NGS? That is the question where this talk lives. The first filter is physical, and the first thing it has to fight is host DNA. When you sequence a clinical sample without preparation, you do not sequence the pathogen, you sequence the patient. In cerebrospinal fluid, abbreviated here CSF, pathogen DNA can be less than 0.1% of total reads. In plasma cell-free DNA, the microbial fraction is typically below 1%. In bronchoalveolar lavage, the picture is variable, often dominated by host signal and oropharyngeal commensals. So the wet lab first job is not to sequence, it's to enrich. Host depletion, microbial enrichment, sometimes selective lysis, these are not optional refinements. These are precondition for everything downstream. For the geneticists in the room, this will sound familiar. The same logic that drives cell-free DNA enrichment is non-invasive prenatal testing, NIPT, and in liquid biopsy, oncology drives host depletion in metagenomics NGS. The disciplines are converging on the same problem. You cannot sequence what you have not first concentrated. The second piece of physical noise is contamination. The most important paper on any new metagenomic NGS lab should read is the 2014 Salter paper from Sanger Institute, which gave us the term chytome. The chytome is the background of microbial DNA already present in your reagents, kits, plasticware, and lab environment before any patient sample touches. Universal, every laboratory has one. The question is whether you have characterized yours. In low biomass sample, the ketone can produce more reads than genuine pathogen. If you are not running negative controls and tracking them across batches, you are not running a clinical assay. You are running an experiment whose floor you cannot see. This one is the harder cultural shift of laboratories coming to metagenomics NGS and the culture-based microbiology background. In culture, contamination is visible. In sequencing, contamination is invisible until you measure it deliberately. The third element of the physical filter, and where I will be most direct, standardization is not a comfort feature. In low biomass diagnostics, technician-to-technician variability is the limit of detection. Extraction yield varies, library preparation efficiency varies, pipetting volumes vary. Each is a small effect. Together, in a sample with very few pathogen reads to begin with, they determine whether the pathogen is detectable on Tuesday and not on Wednesday. A clinical assay whose limit of detection moves between runs cannot, by definition, support clinical reporting. You can publish it, you cannot diagnose with it. Operator-independent, closed workflows exist for one reason. They are the only way to deliver a reproducible limit of detection, and the reproducible limit of detection is the precondition for everything a clinician will eventually act on. Back to our patient. The team has decided to send metagenomics NGS. What does the wet lab now have to deal with? Volume of cerebrospinal fluid, 3 milliliters. The limit of what the lumbar puncture yielded. Pre-analytics already constrained. The sample was drawn approximately 36 hours after empiric antibiotics were started. So whatever organism is causing this illness has been chemically pressured for a day and a half before it arrives at the wet lab. The wet lab makes 3 deliberate decisions. Host depletion, because the 3 milliliters of cerebrospinal fluid dominated by leukocyte DNA will not yield a usable pathogen signal otherwise. Negative controls in parallel, because the ketome of this batch must be visible along the patient's reads. Automated closed-loop library preparation, because this is a clinical sample, not a research sample, and the limit of detection must be reproducible. None of these decisions are visible in the eventual report, but the wet lab has, before the sequencer is even loaded, already determined what is possible to detect. We move to the second filter, the computational one. This is where the dry lab does its work, and the most important thing to say about clinical bioinformatics is that it does not produce a hit. It produces an estimate of a probability that a hit is real. Modern metagenomics NGS pipeline has at minimum 4 serious components. First, human read removal. Sometimes 99% or more of read maps to the host and are discarded. Second, taxonomic classification against the database. Your pipeline is only as good as its reference. A clinical grade pathogen database is curated, version controlled, chosen deliberately. A research grade database dominated by environmental sequences will produce confident looking calls for organisms that are not pathogens. Third, abundance and coverage thresholds. A single read map to Mycobacterium tuberculosis is not tuberculosis. The reads have to cluster, coverage has to be uniform, abundance has to exceed plausible contamination. Fourth, contamination-aware calling. The ketone must be subtracted from the patient call using no template controls or NTCs that travel with each batch. This is non-trivial. When all 4 work together, you get a callable result. When any one fails, you get noise dressed as confidence. If there is one sentence I would like you to take from this talk, is this one. Metagenomics NGS detects nucleic acid, it does not detect disease. This sounds obvious, but it is not. A surprising amount of clinical metagenomic NGS reporting in the published literature and routine practice treats a positive read as a positive diagnosis. It is not. Unorganized nucleic acid can be present in your patient's sample for many reasons. Active infection, colonization, latent reactivation, sample contamination, kit contamination, environmental exposure. The job of the computational filter is to estimate the probability that the sequence you have detected is the agent of the disease in this patient at this moment. That is the entire intellectual content of clinical metagenomics mNGS. Everything else, throughput, cost per genome, depth of coverage, is upstream to this question. The third element of the computational filter is turnaround time, and I would like to argue for a particular framing. Turnaround time, sometimes abbreviated TAT, is not an information technology metric. It's a clinical variable. Consider two metagenomic NGS results. One arrives 24 hours after the sample is drawn. The other arrives 96 hours after the sample is drawn. Both detect the same organism with the same confidence. Are they the same tests? They are not. The first arrived in time to drive initial therapy. The second arrived after the patient had been on empiric coverage for 4 days, and the clinician decision space has collapsed. The second test, even when technically perfect, is a confirmation. The first one is a diagnosis. This is why the choice between targeted panels, full metagenomic NGS, and the hybrid approach is not a technical choice, it's a clinical one. Each maps to a different clinical timeline, not a different ambition. The right test, in my view, is the one that arrives in time to change what the clinician does. Anything later is interesting, only matters, only what arrives on time matters for clinician. Back to our patient, the metagenomic NGS run completed on day 5. The dry lab now has to make sense of what the sequencer saw. The output contains 3 categories of signal: low but consistent reads, mapping to Listeria monocytogenes, reads to 2 organisms also present in ketone control, background humor commensal sequences. The bioinformatics filter discriminates among these using 3 different criteria. Listeria clears the abundance threshold, has a consistent coverage pattern across the genome, and critically is absent from the negative control. It is called a real signal. The 2 ketone organisms are present in the patient sample, but also in the negative control, which means we cannot distinguish patient signal from reagent signal. These are rejected. The commensal background is below the reporting threshold and is not included. 3 different signals, 3 different reasons for the decision, none of them visible in the one-line report. We arrived at the third filter, the cognitive one, and we open with the report. Reports are designed by bioinformaticians for bioinformaticians, fail clinicians. I have read metagenomics NGS reports that listed 40 taxa with read counts in scientific notation with phylogenetic trees attached and a footnote saying clinical correlation recommended. A clinician at 3 in the morning deciding whether to escalate antibiotics cannot use that report. An actionable infectious disease NGS report has, at minimum, a top-line call, a calibrated confidence statement, a list of plausible alternative explanation, an explicit statement of what the report is not claiming, and one named next clinical step. That last element matters. A report that ends without telling the clinician what it suggests they do next is incomplete. The genetics parallel. In 2015, the American College of Medical Genetics, ACMG, and Genomics, together with the Association for Molecular Pathology, issued a joint guidelines that gave us 5-tier classification system. Pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. That paper transformed clinical genetics reporting because it gave every report a shared grammar. Infectious disease NGS does not yet have this equivalent. We are still reporting in dialects. Let me show you what this looks in practice. This is a redacted sample report from our laboratory pipeline, rendering exactly the case we've been discussing. 67 years old with Listeria meningoencephalitis. Notice how these 5 elements I just described actually appear on the page. Top line call, Listeria monocytogenes detected. Calibrated confidence, tier expressed explicitly. With the supporting evidence, abundance, coverage, absence in the negative control laid out so the clinician can see the reasoning, not just the conclusion. Alternative explanation, brief, candid. This is what else might explain the signal and why we ruled it exactly. Explicit what is not. The report calls out the absence of evidence is not evidence of absence. fastidious organism, low biomass viral pathogen, organism below the limit of detection will not appear here. And finally, the next clinical step, continue Listeria-directed therapy. Consider repeat lumbar puncture if the clinical course deviates, multidisciplinary team discussion, what we call an MDT, if antibiotic narrowing is being considered. This is not a marketing slide. This is what the clinician on the ward sees at 3 in the morning. The point I want you to take away is that the report itself is a clinical instrument, and how it is designed determines whether the test changes the decision. When do clinicians actually start to use a new diagnostic test? The empirical answer in my experience is that clinicians use tests they trust. Trust is not built by validation paper. It's built by serial concordance. A clinician orders the new test on cases where they already have a strong suspicion. The test agrees, confidence goes up. Clinician orders tests on cases where suspicion is weaker, the test surprises them, and they investigate. Either outcome calibrates trust. This is why the first 50 cases of any new infectious disease NGS service are not yet routine clinical workflow. They are trust-building exercise. Programs that skip this stage, that try to launch as routine offering on day one on the strength of vendor claims, typically get ignored. They sit unused, the trust was never built. One institutional structure has been shown in oncology genetics to materially change how complex genomic results translate into clinical action, the molecular tumor board. A recurring meeting where a complex case is reviewed jointly by a pathologist, a geneticist, an oncologist, and a bioinformatician. Infectious disease NGS needs the same structure. Call it a molecular infectious disease board. Clinical microbiologist, bioinformatician, infectious disease consultant, and when relevant, a geneticist or immunologist for host factors. The reason this works is not the meeting itself, is that the complex metagenomic NGS reports almost always contain information that is ambiguous to any one specialty alone, but interpretable when several specialties look at it together. Automation enables the meeting, fast reports, clean evidence summaries, easy case packages. Automation does not replace the meeting. The cognitive filter, when it works, is a meeting, not a piece of software. Our patient, one last time, on day 6, the multidisciplinary team meets infectious disease, microbiology, bioinformatician who ran the pipeline, and neurology team caring for the patient. The conversation moved through 3 filters in reverse. The neurologist confirms the clinical course is consistent with bacterial meningoencephalitis. The bioinformatician walks through why the Listeria call is real and why the ketome organisms were rejected. The microbiologist confirms pre-analytics were validated within range. The infectious disease physician accepts the call, narrows therapy to ampicillin monotherapy, discontinuing ceftriaxone, vancomycin, and acyclovir, and plans a 21-day course based on standard guidelines for Listeria central nervous system infection. The patient completes the course and is discharged neurologically intact. The deliverable was a decision. Which antibiotic for how long, with what monitoring? The genome was the input, it was not the decision itself. This is what a clinical metagenomics NGS program looks like when all 3 filters work. The point is not that the sequencer found Listeria, the point is that the system delivered a defensible, narrow therapeutic plan in time to matter. What does the next 5 years look like? A ground forecast, not a utopian one. 4 concrete trajectories. First, sample to answer time. The current state of the art for routine clinical metagenomics NGS is days. The trajectory is towards hours, the single most important variable for clinical adoption. Second, host response transcriptomics. Several groups, including Salik et al. And Katri at Stanford, have shown that host gene expression patterns can distinguish viral from bacterial respiratory infection and stratify sepsis patients. Combining a pathogen call with a host signature gives the clinician something neither alone provides. Third, real-time prediction of antimicrobial resistance, AMR, directly from sequencing data. In drug organism pairs where the genotype-phenotype concordance is well validated. Not every combination, but many. Fourth, non-invasive sampling. My group has been working on britomics, analyzing exhaled breath for respiratory infectious disease application. The next generation of infectious disease diagnostics will not always require a sterile site sample. If I had to give you a scorecard for where the field is, it would look like this. The physical filter, the wet lab is closing fast. Automation, host depletion, ketone management, standardization, all maturing. The computational filter, dry lab is also closing fast. Pipelines maturing, database improving, validation frameworks published. The cognitive filter, the bedside is stuck. We do not have yet shared reporting standards. We do not yet have routine multidisciplinary structures around infectious disease mNGS in most centers. Clinician trust is built by case slowly. Reimbursement is uneven. The structures around the test, not the test itself, are what is rate limiting. This is, in fact, encouraging news. The remaining problems are not technological. They are organizational, and the organizational problems, while harder than they look, are tractable. Let me finish with this. The lab that wins this decade is not the one with the best sequencer, it's the one with the shortest last mile from sequencer to bedside. Thank you very much for your attention. I look forward to the discussion and to learning from the experience of our colleagues in China. 谢谢。